In this video, I'm going to cover transcription and translation, and we're going to try to answer the question, how is the DNA code used to build proteins? And so how does the process of protein synthesis work? It includes two major steps. The first step is transcription, where a gene in DNA is rewritten or transcribed into a molecule called mRNA or messenger RNA. So DNA, which is in the nucleus, is going to go through the process of transcription, and we're going to make RNA, which is single-stranded. In this diagram, that process is being showed in this red area, which is the nucleus of a cell. Transcription occurs in the nucleus because that is where DNA is located. The second step of protein synthesis is translation, where that mRNA that's made in transcription will leave the nucleus, and its code is going to be used to build a protein one amino acid at a time. So translation is simply taking RNA and building a protein, which will have a function in the cell, and that process occurs outside the nucleus at a ribosome. So let's take a look closer at what happens during transcription. So transcription occurs in the nucleus, and during transcription, a protein called RNA polymerase unzips DNA near a gene and uses DNA as a template, as a guide, to build a molecule of mRNA, also known as messenger RNA. And so in this diagram down here, you'll see this kind of brown blob is RNA polymerase. It is a protein whose job it is to find a gene on DNA, which you'll see the DNA is the double-stranded blue molecule here. It's going to unzip it, and it's going to expose the letters on DNA, and those letters are going to be used as a template. And so what's going to happen then is the mRNA that's going to be produced is simply, it represents a rewritten version of the gene so that the DNA can stay in the nucleus and the code for a gene can leave. And so what happens during transcription is RNA polymerase is going to be moving from left to right in this diagram, and RNA polymerase is going to be using the letters on DNA, and it's going to be using it as a template. So if DNA has a T, it is going to pair a complementary A with that T. And if DNA has an A, RNA polymerase will pair a complementary U. Normally it would be a T, but remember, if we're building RNA, which is what this red molecule is, we use U, uracil, instead of T. Right? And it's going to do this letter by letter. A G will be paired up with a C, another G paired up with a C, and you can see mRNA is being built one letter or one nucleotide at a time. Okay, so that is transcription. This mRNA that will be built is going to leave, and we're going to then do the process of translation. And so once the mRNA leaves the nucleus, it travels to a ribosome where it is translated. This is typically in the cytoplasm or a place called the endoplasmic reticulum. The mRNA code is going to be translated one codon at a time. A codon is simply 3 letters of mRNA code. And so if I look at this diagram, I see the mRNA is this kind of red strand down here. And you'll notice that in this diagram, it separates this mRNA code into triplets, or a series of 3 letters that we call codons. And so what's going to happen is these codons are going to be read one at a time. And a molecule called tRNA, transfer RNA, carries a specific amino acid. Those are the building blocks of proteins, and matches those the tRNAs with a codon on the mRNA. Each new tRNA molecule adds an amino acid to a growing chain, which will eventually fold into a protein. And translation is going to start at what we call a start codon, and it's going to end at a stop codon. So let's look back at this diagram. We'll see there was a codon here that was AGU, and you'll notice there was a tRNA that paired with AGU because it had what we call an anticodon that is complementary, or the opposite of the codon. There's another tRNA here that is pairing with CGU because it has an anticodon that matches with it, and notice it brought with it this little purple circle which represents an amino acid. And there's another tRNA behind that, once again, we see that pairing, and it's bringing another amino acid. Those amino acids will be joined together by a bond, and what we end up creating is this chain here that I'm highlighting in yellow. This chain will eventually fold once you get a bunch of amino acids into a complex shape, and then you will get a functional protein. And so that is the process of translation. And to do this process of translation, we're going to use a codon wheel to identify the amino acids that are being built. So let's do that. So if you were asked to transcribe and translate a gene, you would take a section of DNA, a gene, and the first step is going to be taking the DNA, and through the process of transcription, we're going to make mRNA. And you do that by thinking about what would pair with each letter on the DNA. Now T pairs with A pairs with U in RNA, C pairs with G, and I would keep doing this transcription, and I would get the following code. And so my mRNA, you'll notice, is I wrote it in these triplets, in these codons. And what I'm going to do now is I'm going to look at each codon one by one, and I'm going to translate it. And so going from the mRNA to amino acid, this is translation. And to do this, I'm going to use a codon chart. So AUG is my first codon. And so I'm going to use this codon wheel. And you'll notice that this codon wheel has a center circle with a G, U, A, and C. Then there's a second circle, and then there's a third circle for the three letters in our codon. The outermost ring is all of the 20 different amino acids that those codons can translate into. And so the way to do this is you start in the center and work your way out. So if my first codon is AUG, then I'm going to start in the middle, A. I'm going to work out to the next row, U, and then the third row, G, is MET, or methionine, which we see is actually a start codon. So what I get here is I get a start, which technically is MET, which stands for methionine, which is an amino acid. Okay, the next codon, GAA, same idea. Start in the middle, GAA, I get GLU, my next amino acid, GLU. And the next codon then, UUC, I see is PHE. My next codon, AUC is ILE. And the last one, UAA, I notice is a stop codon. And so you'll see this protein starts with a start codon, and then I have 1, 2, 3 amino acids, and then a stop codon. Now this is a 4 amino acid protein. Most proteins are hundreds or thousands of amino acids long. And so do all cells produce the same proteins? So the process of protein synthesis is regulated or controlled in many different ways. And so in some cells, genes are switched on. And in other cells, the genes might be switched off. And so the proteins that are made by a cell will be based on the function that a cell has. So for example, we all started out as one cell, and those cells have not decided what type of cell they're going to be. And during development, you'll produce muscle cells, fat cells, bone cells, blood cells, right? All these different types of cells. Now, something like hemoglobin, a protein that's used to carry oxygen, you would find hemoglobin in blood cells. But you wouldn't necessarily find hemoglobin in these other cells because their function isn't to carry oxygen like blood cells are. And so what you see is that depending on what the cell's job is, the types of genes that are turned on are going to be different, which means the types of proteins that they make are going to be different. And so that leads to cells with very different jobs based on the different proteins that they contain. And that's it. Thank you for watching.